postdoc at Florida State University working with Dr. Kim Hughes, and going to be presenting a project asking why it is that female Trinidadian guppies exhibit a mating preference for unusual looking males. Natural populations are often found to have high levels of genetic variation in ecologically important traits, and this genetic diversity has important implications for disease resistance, for the ability of populations to respond to environmental change, and by consequence for population persistence. And for these reasons, understanding the processes that maintains genetic variation is fundamentally important in ecology and evolutionary biology. One process that can maintain genetic variation is negative frequency dependent selection. And this can occur when the fitness of a phenotype is inversely related to its frequency in the population. Because rare alleles are favored, they're unlikely to be lost from the population, and so this can maintain genetic variation over evolutionary timescales. Examples of negative frequency dependent selection have been found in a handful of different systems. Some classic examples include flower coloration in the elder-colored orchid, the different color morphs of five-blotched lizards, and shell polymorphism in grove snails. And in light of this evidence, there's been much interest in determining what sorts of ecological mechanisms can generate negative frequency dependent selection. And one such mechanism is a rare male mating advantage, and this can occur if females exhibit a mating preference for rare or novel phenotypes. And this kind of a mating preference for novelty has been observed in 3 different species of ciliate fishes, in fruit flies, and there's some evidence in humans as well. But despite this evidence, it's not clear why a mating preference for novelty exists. It's not known what behavioral processes underpin this mating preference for novelty, and it's also not clear what the evolutionary origins of this preference may be. And I think that these are important questions to address, both to better understand the preference itself, and because answering these questions will help shed light on the potential generality of this preference as a force maintaining genetic variation. All right, so to help address these questions, I wanted to test one potential mechanism that could be responsible for a mating preference for novelty. And specifically, I hypothesized that this preference could result from habituation. So habituation is the process by which an organism becomes less and less responsive to a repetitive stimulus. For example, when the air conditioning unit in your office first comes on, the noise is probably really noticeable at first, but as it continues to drone on, you gradually stop noticing it. And this is because of changes in the central nervous system that cause you to tune out repetitive information without even realizing that you're doing it. And generally speaking, habituation is advantageous because repetitive stimuli tend to be unimportant, and by filtering them out, that frees up our attention to focus on other more relevant stimuli. If a particular sexual signal is common in the population, females should encounter it repeatedly, causing their interest in that signal to wane over time. And all else being equal, this means that a common signal should be less appealing than a rare or novel one that females will be encountering for the first time. And so if females do habituate to male sexual signals, this could explain a mating preference for rare or novel signals. And indeed, there is some evidence that females habituate to sexual signals. In common grackles and zebra finches, there's evidence that females prefer more complex songs, and this is thought to be because females habituate to simple repetitive songs. But to my knowledge, no previous study has asked whether habituation can cause a mating preference for novel phenotypes. And this is an important distinction because it's a preference for novelty that can generate negative frequency dependent selection and thus maintain genetic variation. One of the reasons I think that this is an interesting question to address is that habituation is a pervasive feature of animal sensory systems. It's been found in virtually every animal species that's been tested, and habituation is known to affect responsiveness to a pretty broad range of stimuli. And so if habituation is responsible for a mating preference for novelty, that would suggest that such a preference could be widespread. So to test whether females habituate to male sexual signals, I chose to use the Trinidadian guppy as my model system. The color patterns of male guppies are both diverse and heritable, providing one of the most extreme examples of morphological polymorphism. And this polymorphism is thought to be maintained, at least in part, by the strong mating preferences that female guppies show for males with rare or novel patterns. All right. And to test whether female guppies are habituating to male color patterns, I wanted to test 4 key criteria that psychologists commonly use to demonstrate habituation. And so if a response exhibits these 4 criteria, that's considered to be diagnostic of habituation. And I'll walk you through what each of these criteria means and how I tested them. So the first criterion on the list is responsiveness decline, and this just refers to the reduction in responsiveness that's caused by repeated exposure to a particular stimulus. To test for responsiveness decline, I first wanted to provide a baseline measure of female mating interest, and I did this by taking a group of females that had never been exposed to any male color patterns and assaying their level of mating interest in a single single test male. So male guppies will show off their color patterns to females using courtship displays, as shown in the image here, and I scored female mating interest as the proportion of those displays that the female responded positively to. And I'm happy to answer any questions you might have about the behavioral protocols involved. And so this level of mating interest exhibited by these naive females provides basically a point of reference against which to look for a responsiveness decline. And I had a sample size of 20 females per treatment for this treatment and each of the others that I'll be showing. And then to test for a responsiveness decline, I took a new group of females and I visually exposed them to a series of males that all had the same color pattern as one another. And I'm using the color of the male symbol here to indicate whether or not males looked alike. And then following these exposures, I immediately measured female mating interest in a similar looking male. When I was performing the exposures, I showed females one male at a time for 2 minutes each, and I varied the total number of males that they were exposed to. And what I found was that female mating interest declined significantly as the number of exposures increased. And post hoc tests indicated that it took exposure to just 6 males, that's a total of 12 minutes of total exposure, to elicit a significant reduction in female mating interest. So this provides an example of rapid plasticity in female mate choice, and it demonstrates responsiveness decline. All right, and then the second criterion I wanted to test is something called spontaneous recovery. And so spontaneous recovery occurs following responsiveness decline when you isolate the individual from the stimulus, and that causes its responsiveness to recover again. So for example, if you had gotten used to the sound of the air conditioning unit and then it turned off briefly, it would be really noticeable once it turns back on. To test for spontaneous recovery, I followed similar steps as before. So I took a new group of females, I visually exposed them to a series of 15 similar looking males and tested their mating interest in a similar looking male. But the difference was that this time I isolated females for 30 minutes in between exposure and testing. And what I found was that the females who had been isolated prior to testing showed greater mating interest than the females who had been tested immediately. And this provides evidence of spontaneous recovery. All right, and then the third criterion I wanted to test is something known as dishabituation. So dishabituation occurs following responsiveness decline when exposure to a novel stimulus causes interest in the original stimulus to recover again. So for example, if you had gotten used to the sound of the air conditioning unit, but then you heard the siren from a passing police car, that would cause the air conditioning unit to become noticeable again. To test for dishabituation, I again followed similar steps as before. So I took a new group of females, exposed them to 15 similar looking males, and measured their mating interest in a familiar looking male. But the difference this time was that I exposed females to a novel looking male in between exposure and testing. And what I found was that females who'd been shown this novel-looking male prior to testing showed greater mating interest than the females who had not seen a novel male prior to testing. And this is what we would expect as a result of dishabituation. All right, and then the fourth and final criterion to test is something known as stimulus specificity. And this is the idea that exposure to a particular stimulus should not affect responsiveness to a different kind of stimulus. So for example, getting used to the sound of the air conditioning unit would not make you any less likely to notice if the fire alarm were to go off, which is something that we all experienced yesterday. All right, so to test for stimulus specificity, I again followed similar initial steps. So I took a new group of females, exposed them to a series of 15 similar looking males, but the difference was that this time I measured their mating interest in a novel looking male. And what I found was that females who were tested with the novel looking male showed greater mating interest than the females who'd been tested with familiar looking males. And I also found that the level of mating interest females showed in the novel looking males was not significantly different from the level of mating interest that the naive females had shown. And what both of these results indicate is that exposure to a particular color pattern does not reduce interest in a different type of color pattern. And this is exactly what we would expect as a result of stimulus specificity. So collectively, these results satisfy the 4 different criteria that are commonly used to demonstrate habituation. And this tells us that habituation underpins a mating preference for novel phenotypes in the Trinidadian guppy. And this is exciting because it provides a mechanistic explanation for this mating preference, which has been observed in the guppy and a handful of other systems. One of the reasons I think this is an interesting result is that habituation is a very general property of animal sensory systems, and so habituation probably plays a broad role in animal mate choice. And in combination with my results, this suggests that a mating preference for novelty could be widespread. Tests of a mating preference for novelty are still relatively rare, and so it's possible that this kind of a preference could play a broad but underappreciated role in the maintenance of genetic diversity. My results also provide some clues as to the evolutionary origins of mating preference for novelty. The environment is constantly bombarding us with sensory information, much of which is repetitive and unimportant, and habituation is thought to have been favored by selection because it helps us to filter out repetitive information, freeing up our attention to focus on other stimuli that are more relevant to tasks like locating a food source or detecting the image of an approaching predator. And so one potential explanation for a mating preference for novelty is that it could be an evolutionary byproduct of selection favoring habituation in non-sexual contexts. This is essentially a version of the sensory bias model of mate choice evolution, but the idea of a sensory bias for novelty is not something that's previously been tested. And so this is an idea that I'll be exploring in greater detail in my future work. So with that, if you're interested in learning more about this project, it recently came out in Proc B, so please look it up. And I'd like to thank the funding sources that supported this work, including the Dean's Postdoctoral Fellowship at FSU and NSF. And I'd like to thank a number of individuals that provided feedback on my talk, on my science, or who helped to maintain my many fish. And I'm happy to take any questions.
Yeah, nice talk, Mitch. Any sense of habituation for other things that are like the male guppy, right? So like a color ball or something that she would normally be attracted to. Right. So does habituation to fish like desensitize you to novel objects?
Does habituation desensitize you to novel objects? Yeah, so habituation is quite a general property. Organisms don't habituate to everything, but they habituate to a lot of different things. So my expectation, if you were ever to test them with a new type of stimulus, is that they probably will habituate, especially if that cue is not sort of reinforced with something that's biologically relevant to them. Does that answer your question?
Yeah, I was thinking if you're surrounded by like red berries, and there's no red males, and a red male comes in, you become like you wouldn't find him novel because you're habituated to the color red.
That's right. Yeah. Yeah. Although maybe one piece relevant to that is that in this case it appears that the color pattern itself, rather than a particular cue, is what they're habituating to. So maybe seeing red in combination with all the other features of the male could be novel in a sort of a combinatorial way.
Yeah.
Do you think males would habituate to non-receptive females?
Males would habituate to non-receptive females. I suppose, yeah, if there's like a particular cue that's signaling that a female's non-receptive, would they habituate to that?
It's maybe more of a thought question. I don't know. I was just curious.
I guess it would depend on how females are signaling that they're non-receptive and what cues are involved in that. And of course, habituation isn't the only form of learning that males are capable of engaging in. So I think it's quite possible that if a female is indicating she's non-receptive, they're probably able to learn what that feedback means and